A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Chief Operating Officer of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about topics relating to antibody discovery and development. Our expert speaker, Dr. Jesper Sorensen, will discuss 3D antibody modeling in Orion. Please note the webinar is being recorded. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I'll now turn the show over to our speaker.
Hey, Janice, thank you so much for the invitation to come speak here at the Antibody Society, and thank you for the Antibody Society for the invitation. It's fun to talk about something new and interesting in Orion for us, and so I'm going to talk a little bit about how we're doing antibody modeling in Orion. But first, I'm just going to introduce OpenEye because I don't know that everybody in the audience knows OpenEye. So sort of this is my at-a-glance slide. We've been in the scientific and computational modeling business for 25 plus years, 26 plus years, it says here, and really pioneered Orion, which is our cloud-native molecular design platform. Like I said, we've been in the business since 1997. We never took any venture capital money, and we're currently rounding around 170 employees. But we were acquired by Cadence Design Systems about a year and a half ago now, which has sort of enabled us to expand beyond what we historically have been offering. So I'll talk a little bit about Orion. I'll talk a little bit about our general offerings as well. So what we do is we develop computational software for the pharmaceutical and biotech industry. We offer sort of 4 different areas of support or software. We do consulting services with our software. We also develop toolkits. This is sort of how do computers understand molecules and protein structure and things like that. We develop those into desktop and desktop applications on Windows, Linux, and Mac. And we also have a web-based cloud platform called Orion. And I'll talk more about that in a minute. One of the reasons I, want to just mention our applications and toolkits is the toolkits are really the foundation of everything we do at OpenEye. Even things that are in the cloud really come from the foundation of these toolkits. And so we ship 19 toolkits in 14 different applications. The 19 toolkits span everything from cheminformatics to virtual screening, protein structure preparation, and getting structures ready for simulation. So it's a wide array of search tools and APIs that people can access and use. They're actually free to academic labs. So in some ways, it's a fantastic offering. And what I'll talk to you about today is really built upon those toolkits. So Orion is our cloud platform. So I'm sort of showing you the bottom of this. The bottom graph here is the Orion. It's the platform. It's the web interface. It's where you can create data, analyze data, do some serious calculations, and also collaborate with your team. You can share your results. You can communicate with graphs and slides and 3D images. You can save things and say, hey, colleague, look at what I just, look at these interactions, and you can share that image and they can go straight to it. So that's sort of the platform. That's where you log in and you can see all your data and you can interact with it. But on top of the platform itself, it's really sort of our scientific turnkey solutions. And so we have a small molecule discovery suite. That's probably our most expansive because that's the origin of OpenEye, is really ligand-based and structure-based discovery solutions. The antibody discovery suite is relatively new. We have a formulation suite as well, which is sort of at the end of small molecule drug discovery, when you want to start formulating your drug into a pill form or something along those lines. That's really where we have specific tools for that. We have third-party modules as well. We've integrated with Gaussian for QM calculations. We have additional third-party software, either something we put in or other people have put in. And you could actually put your own in-house tools in as well. So Orion is really sort of very expansive of what you can do today. And I'll talk more in detail as we get going about what we put into Orion and specifically the antibody modeling suite. This is the Antibody Society, so I really should not introduce antibodies. Probably shouldn't have to, but at the same time, it's always nice to get on the same page about what we're talking about. And so antibodies are these large proteins in your body with incredible diversity. They're produced by the B cells of the immune system to protect against pathogens, and they're the most diverse protein in your body. And millions of these antibodies circulate in your body at any given time. And so, if we just take sort of the canonical example of an antibody, IgG, you can see this sort of Y-like structure. It doesn't actually look like that necessarily in real life, but at least schematically, this is a really nice figure of what an antibody looks like. And you have this FC region, the effector region, and then you have the Fab and specifically the FV region up here. And at the tip of the FV region, you have these CDRs, these complementary-determining loops. These are really the loops that depending on their composition will bind to different targets, different protein targets for the pathogens or the antigens that they want to connect to. You can see them in sort of different representations here. I kind of liked all these, and I thought they were great illustrations of what an antibody looked like. So I won't really go into like where all this diversity comes from. It's a gene recombination event that really creates all this incredible diversity, and then there's somatic mutations after those events as well. And Andrew Bradbury from Specifica Bio, who we collaborate with, gave an incredible masterclass on this. It's on their website, and I would recommend it. It's many hours of learning about antibodies, and I'll just give a shout out to Andrew and say this was an amazing class, and everybody should probably go take it if they haven't already. But so what I think the key point here is there's a theoretical max for how many different antibodies we think can be produced, and the number itself is potentially up for debate, but it's somewhere in the 10 to the 18th or 10 to the 20th. It's really an incredibly high diversity in what an antibody structure can look like. To give you some sort of correspondence, for small molecule drug discovery, we believe that number is about 10 to the 60th. So that's so much higher, but at the same time, that doesn't mean that either of these two problems are easy just because it's smaller. And I think one key thing I want to just mention, of course, is that most of this diversity in these antibodies really comes in the 8-CDR3 region. That's the CDR3 on the heavy chain. That's really where most of the diversity comes from, and where most focus for antibody drug discovery is focused. That doesn't mean there's not diversity elsewhere, but that's really where the majority is coming from. So the first product in our antibody discovery suite came in 2021 with AbXtract. This is an offering we did in collaboration with Specifica Bio, and it's really about analyzing next generation sequencing data. And with their tools, whether you're getting your antibody population from animal immunization or display technologies, using AbXtract and next generation sequencing, you can really get a much higher set of antibody leads from your sort of panning and discovery campaign. And those antibody leads will have greater functional diversity, broader affinity ranges, and fewer sequence liabilities. Already sort of built into the analysis tools there. And if we sort of look at the drug discovery pipeline for antibodies, and I really like this figure from Saying It All, and I modified it slightly just to have a little less detail. But if we think about where AbXtract comes in, it's sort of in the antibody generation and early antibody optimization stages. And it's already been quite helpful in moving discovery campaigns forward in this aspect. But what we're adding today, or what we're adding now, is effectively more tools in the antibody optimization area. And so these are the tools we're adding now, is 3D, is sort of adding 3D to the existing sequence-based tools. So we're adding structure generation and preparation. We're adding property calculations, structural ensemble generation, some structure-based clustering that I think is new and interesting to people in this field, and structural modifications as well. So we're really building on top of what AbXtract already set out to do for our antibody discovery suite. So I'll talk about each of these 5 points in sort of different vignettes. And the first here is structure generation and preparation. And so we'll go into that. One too many. Okay, so the basic tooling here is you have a sequence. You could have gotten it from OAS. You could have gotten it from your internal discovery campaign. You could have done it with, this could have been your 100 or 1,000 sequence outputs from AbXtract analysis. And we can generate structures of those, and we can do structure preparation and some structural annotations. And currently we're using AbodyBuilder 2 or ImmuneBuilder from Charlotte Dean's lab at University of Oxford. I put the citation here at the bottom. I'm sure most people here are familiar with it. What we then generate is a design unit. It's a data structure we built at OpenEye, originally intended for small molecule protein ligand drug discovery, but it fits pretty well here as well. So we're marking the binding site residues as CDR3, and we're coloring and labeling the sequences and the regions, and you can sort of see in these 2 images just what that looks like. The purple here is HCDR3, and you can see I have labels, and I also have some other annotations I'll get into in a minute, because one of the things we've also done is biophysical liability annotations. And those annotations are historically sequence-based, saying, oh, are these sequence patterns in the CDR regions, and we'll flag them. But what we've done, on top of just saying they're sequence-based, we've also made sure that we only flag a specific sequence if it's actually surface exposed. So it sort of denoises some of those things a little bit to what we think are relevant liability annotations. This is where structure really kind of comes into play. The other thing we've done is some basic tooling around preparation of protein crystal structures, or not crystal structures necessarily, could be cryo-EM or NMR as well. But this is really built into our OpenEye Spruce toolkit, which we use for all protein structure preparation for docking, MD, or anything else. We're using it here for antibodies as well. So one of the things it does, of course, is it takes an experimental structure and it prepares it, adds hydrogens, and it builds regions that are currently not built out. And so one of the things it also does is sort of structural classification. So here it's highlighted the FV region, and then it's also highlighted the antigen, and it can recognize that because it's using some sequence detection patterns to figure out what's the antibody and what's not the antibody. So it can categorize the antigen into a separate region here as well. And you can see, I don't know, it's a little hard to see on this picture, but on the antigen itself, we've actually built out a loop, and it's colored brown. And that's because it wasn't in the crystal structure. But if you're going to do any type of simulation, you probably want that loop there. It was flexible in the structure determination, so they couldn't place it necessarily. But for simulation, you probably want it there to let it be flexible. And so we build those things in as part of the Spruce preparation pipeline. The other thing we do is we mark crystal packing residues. And I think it's probably too hard to see here, and I apologize for that, but there's purple residues out here sort of floating in space. Those come from crystal packing, crystal cell packing, effectively, especially when you have crystal structures that you've gotten from X-ray crystallography. I think I have a better example of that here in a minute, because in this case, those packing residues don't actually interfere with the antibody-antigen binding interface. But if you're looking to, here's the other structure, 6OE4. It doesn't have an antigen, but as you can see, we've classified the FV region. We can turn on and off water, so you can see the waters around the FV region. But also you can see here on this last image, a lot of the purple packing residues from the crystal experiment. And part of the reason this is important is because if you're trying to say, let's say we're using AI to fold this sequence into a structure, and you're saying, well, it should mimic the crystal structure as best possible. That's our metric of quality. And I understand that metric, but you have to understand that the way those CDR regions or those loops are packed at the interface in a case like this is really strongly determined by the crystal packing experiment. So once you get that out in solution, that may not actually be what those loops will look like. And I think that's an important distinction we have to recognize when we do quality assessment of, can we get an AI or other types of folding mechanisms to fold these into protein structures that we think have relevant configurations. Well, what are we holding them against, right? Because we're holding them against something that has crystal artifacts or crystal packing artifacts that's maybe not completely fair. And I think sometimes when we do quality estimation, we miss that point. But Spruce will show you that. And in Orion, and even in our desktop tools, you can see packing residues and you can see the effect of them on these different configurations. We've implemented basic numbering schemes, just 4 at this point. We probably expanded to the more complete list. We're using Anarchy from Dunbar and Dean. You can see 4 different coloring schemes here: IMGT, Kabat, Kotia, and Martin. And we also have sequence viewer as well, of course, and you can see those same color patterns that you're seeing in 3D are also demonstrated in the 2D. So it's really easy to sort of use the sequence along with the structure to get into that. So one of the things that's sort of interesting to us, because Orion is really a fantastic tool to scale, to do things at really high scale, is how does this perform in Orion when we're doing AI-based folding with ImmuneBuilder? And really, once there's a little bit of a startup cost because we're moving model files around in the cloud, but once we start and we run this at scale, we can actually take sequences and fold them into structures or models at about 0.1 second per structure. And the cost of doing that in the cloud is, $0.003 to $0.004 per structure or per sequence at scale. And I think just to sort of highlight the comparison here is we could do the human paired sequences of FV and the OAS. And there's, I think, when I checked, is probably like 1.7 to 1.8 million different paired sequences. And we projected we could actually generate structures of all of those for about $5,000 to $7,000. And I think there's ways to make that even cheaper and more performant. But at some point, you have to decide to stop over engineering something or over engineering something to make it even cheaper. But I think that we could make it cheaper if that's interesting. But you can sort of see that, the total number of structures and the total time, it kind of keeps going up, which you would expect, but it scales relatively linearly. The 2 different sort of points here are how many CPU instances I'm allowing the Orion to scale out to. So if I say you can only have 1,000 instances max, effectively at some point you sort of plateau. If I let it have 10,000 instances, you can see my total time at some point starts actually going down, and I start saturating those instances. Of course, using 10,000 instances on Orion also adds a little bit of cost, and so that's where you can see, the cost per structure goes up a little bit as I scale out. But I think we can debate these numbers, but I think they're pretty solid, and the cost performance here is pretty solid. So moving on to property calculations, sort of thinking about developability long term. So there are already some really nice papers out there sort of talking about different characteristics of antibodies, the properties. That's work both from Matt Rabel and team with Charlotte Dean at University of Oxford. And then there's some really nice papers sort of, I won't call it follow on, but because they have very different sort of properties they look at, but from Boehringer Ingelheim in 2021, and I put both references here. What they're looking at, you'll notice that they both have 5 metrics, if we can call them that. And that it's sort of meant to mimic, I think, Lipinski's rule of 5 that we know from small molecule drug discovery. And that's maybe 20, 30 years old when, those sort of rules of thumb. And I don't think necessarily antibody drug discovery is 20 years behind small molecule drug discovery, but it's interesting to sort of see these rules of thumb come out of where we can say, what does a therapeutic antibody look like? and if you want to sort of follow in that area, here are some basic properties you can look at. I like these properties, but the ones on the left are sort of very intuitively explainable. The ones on the right are more complicated, but probably also capture some more deeper understanding of the physics involved. I think I'll just show the one graph from that, where you sort of say this is profiling therapeutic antibodies, and where you can see where those antibodies distinguish themselves from a set of known human antibodies. And you can see, for example, the CDR length, or this is the hydrophobicity scores in the blue. Those are therapeutic antibodies versus human, a set of human, which are red. And you can see that, human antibodies have a wider spread of hydrophobicity scores. In the same way, if we look at CDR length, therapeutic antibodies generally have shorter CDRs in general, CDR3 in particular, than known human antibodies. And so there's this interesting distinction between what a therapeutic antibody looks like and what a human antibody looks like. And that, of course, comes down to the body can produce it in the quantities it needs, and distribute it all over your body as needed. But as you move to, we made it in the lab and we have to put it in a vial or in a bag, and it has to sit on the shelf for a number of years, we can't have it aggregating or precipitating out of the solution in that time frame until it can be dosed to a patient. So there are very different properties that are relevant for a therapeutic antibody than there really is for a human, naturally produced antibody. And that's, I think, what you can see in all of these graphs. So I'm just not going to go into detail on any of these, but I think, some of the properties are roughly the same, but some of them really have distinct patterns that I think speak to what the requirements are for a therapeutic antibody. But one of the things I think is interesting is sort of this like hydrophobicity scale, because hydrophobicity, of course, is really important for aggregation and other types of properties. But the way we look at hydrophobicity, I think, varies quite a bit, right? We can do a sort of very naive thing, and so here's an example antibody. You can see I have the antibody here in structure. I have an uncolored molecular surface here on the right, and I can do chemistry-based coloring, right? I can color by aspartate and glutamate, asparagine and aspartate and glutamate, and you can see that I have those in red here. And these two schemes are not too distinct. There's some finer tuned variation between them. But what this doesn't account for necessarily is whether those side chains are actually surface exposed or whether they're pointing to the interior of the protein. So very simple sort of color schemes like this are in some ways not helpful, because yes, there's an aspartate there and that could be a problem, but at the same time, if it's pointing to the interior of the protein, maybe it doesn't matter for hydrophobicity or hydrophilicity. And so I think that those are some of the distinctions I want to make sure we capture. And if we just look straight up at what is the amino acid and not where is it in 3D, that becomes then we have this gap. So some of the things we put into our tooling here is again, we have this antibody again, but now I'm going to show you a grasp surface. That's a very coarse representation of electrostatics, something that Anthony Nichols, our leader, has developed many years ago, and we've sort of now formalized. And you can see where the sort of electrostatic surfaces, positive and negative, are. We have a SAP calculation as well. That's a Poisson-Boltzmann calculation. It's a little more looking at partial charges instead of formal charges. So the picture gets a little murkier, but again, you can see polar and non-polar interactions. And lastly, I'm looking at a SHMAP map. And I'm not going to get into the SHMAP technology, but I'll just say in sort of aggregate is what SHMAP tries to identify is where does a water want to be on the protein surface. And so we've done in-place water calculations on the entire protein surface. It's a great many calculations, but at the same time, it's a great way to look at where does water want to be, because that's maybe a better expression of hydrophobicity and hydrophilicity. And so we're still developing So the technologies we have, that's partly why I mentioned the toolkits early on, we have the technologies to make all these maps or all these sort of surface representations. And now we're trying to figure out how do we translate these into models that can predict aggregation, for example. Ensemble generation is next. So as you know, we can generate structure now with AI or in machine learning. We can even, we could do some grafting based on crystal structures if we really wanted to. But I still think a lot of our goals are beyond just the single structure. We all know proteins are flexible. We expect them to be flexible. So we really want to go beyond just a single structure. So I'm going to show you a couple examples of how we do ensemble generation in Orion and some of the tools we have as offerings there today. So the first ensemble generation we have is loop generation. So this is, again, leveraging that technology we built for Spruce, where when we prepare a protein structure and it's missing a loop of whatever loop it is, we go into sort of a knowledge base lookup and say, what could this look like if it has this sequence and it has this a distance between the points that are missing. And we take that same technology here and we say, hey, can I get 10 examples of HCDR3? And so it looks up in its database, in its knowledge base, and says, here are 10 reasonable examples of what that could look like, and it minimizes them in C2. So that's one way to just say, I just want one loop sampled in different configurations. And it'll show you the energetics as well, so you can rank them if you really care. The other one is MD simulations, of course. And so one of the things we've built in over the years to small molecule discovery suite is this thing we call short trajectory MD. It's really sort of saying, let's just relax these structures and make sure they still make sense. And so again, we do 10 nanoseconds. For small molecules, we probably do only 2 nanoseconds is really generally enough to estimate whether a small molecule will move or not. But for antibodies, we know, especially coming from a machine learning folded structure, we want to do some more simulation and relaxation. So we currently simulate for about 10 nanoseconds. And if you look at the backbone RMSD, you can see it sort of comes up and becomes relatively stable here. And then you can do different selections for clustering. HCDR3 is our default, but you can cluster across all the CDRs in aggregate or independently. And you do an RMSF plot here as well, where you can see we've colored the CDR regions, and of course HCDR3 is where you see a lot of the variability. And you can and at the end of the simulation, we also cluster that simulation into different configurations, and you can see here I have 3 different clusters that it sort of progresses through as it goes through the MD simulation. And this is important because this is really through even 10 nanoseconds of MD, you can see different configurations of those amino acids. And I'll just show you here sort of the yellow was the machine learning folded structure here at the bottom. And then the purple here is 3 different cluster centroids from MD. And you can see they're very different from the AI folded structure. So they've relaxed to a different configuration because they're in solution. The cluster configurations here actually look relatively close to each other. And that's mainly because there's sidechain differences that cause them to fold into different clusters. And there are differences in the structures as well. What I think is really important is, how does this translate into differences in the electrostatics? Because what's on the surface changes as these flexible residues move around. And so as a quick example, I'm just going to show you that the bottom right here was the AI folded structure, machine learning folded structure. And these 3 are 3 different clusters from that MD simulation. These are cluster centroids from that MD simulation. You can see the electrostatic surface using GRASP here is actually starkly different between these 3. Of course, there are similarities, as you would expect. But even then, there are differences in this electrostatic surface. And then you'd think in biology and even in vivo and in vitro, those dynamic differences in electrostatics are going to end up mattering. And that's why we're really keen on using ensemble properties in model building and other avenues. So that's just basic 10 nanoseconds of simulation. But one of the technologies we've built in Orion is protein sampling. And that's for a lot of different reasons. Cryptic pocket detection is another one that we use really sort of enhanced sampling for. And so I'm going to show you an enhanced sampling example here, where what we do is we do normal mode, we do a short simulation, and then we calculate normal modes on the protein, on the antibody protein. And we've focused here on the normal modes around the CDRs. So instead of doing normal modes on the entire protein, in this example here, I'm just showing normal modes around the CDRs, the CDR residues. And we use the modes that are most collective. And I'm not going to get into the details of that in the interest of time, but I just want to put that out there. And it's something I'm happy to follow up on in conversation. But when you look at then what we simulate here, so you'll see as I simulate, I can go along these two normal modes that I picked, and you can see different energy wells. And you can see I start here and I do sample an interesting configuration here, the sort of, moving away from the starting configuration, but still staying in sort of a low energy well. And then as I keep going out, I find, I can keep going up in energy potential landscape. But you can also see, if you look at the RMSD, and this is, if I select on the CDR loops, you can see the RMSD, it's actually still relatively low in the blue here, and then starts getting high as we go up here. If you look at the number of contact fraction, the native contact fraction, when I start the simulation is of course high. It stays high for quite a bit, and then as I move away from that original configuration, I start losing some of those native contacts, and that's desired here. We're trying to explore the sort of flexible landscape of the CDRs, and I have the same two graphs over here with where I've selected on all the, not just on the CDRs, but on all the residues. And I'll just say we can actually do this on Orion at some really interesting scale. And so in this example here, I picked up 3.5 microseconds of simulation in just 22 hours. And it's really an interesting technology to think about if we really want to explore more dynamics of the CDR regions. I'll move on to structure-based clustering. I'm going to talk about a technology we call SiteHopper. And so this was really a technology we built for looking at protein pockets. And we're using our ROX technology that we used for small molecule overlays. We've used it for many, many years. And so we build an image of the active site, protein ligand active site, and we have sort of the surface discriminated here, and then we have color features. You can think of them as pharmacophores if you want to, and sort of color features of the amino acids that line that pocket. And so you can see an illustration of what that looks like here on the right. And the way we then do that is you take a known set of protein structures and you build out these binding sites, and then you can take a query. So I have a query patch here, and I can look at a massive database of patches, and I can say, if I overlay those to the center of mass and I do shape and color alignment, and then I can pull out protein structures that have pockets that are similar. For small molecules, this is really important for things like off-target detection, because it could be that, now I have a drug to target, it's really interesting and it's really cool, but I want to see what liabilities I potentially have. Are there other protein pockets that look like this that I have to be worried about? So it's an interesting technology, and we've taken this technology and we've applied it to antibodies. And so we take the CDR surface and we define those as SiteHopper patches. Again, we take the surface of that, of the antibodies, and then we color them by what the chemical features of the residues at the surface are. And so I'm just showing 4 different surface patches here. And what's really interesting with a structure-based clustering method like this is it doesn't care what the sequence is. There's no limitation that I think I've seen some really nice clustering methods, antibodies, but they all somehow aggregate and say, well, the CDRs have to have the same length for us to cluster them. So we'll cluster all the CDRs that have 8 CDR3s that have 20 amino acids, because that's the only way we can really do sequence-based clustering. And this doesn't care. It just looks at the surface properties and and what the surface looks like. And so this could cluster two different surfaces that have incredibly different lengths of CDRs and even different CDR compositions and say, well, these actually look the same from sort of, if you think of, hey, I'm an antigen and can I bind here? They can be incredibly different. Part of the reason we came up with this method or thought about this method is our collaboration with Specifica, where they have some interesting SARS-CoV-2 data where they have very distinctly different antibodies that all bind the same epitope. And in some ways, we wanted to figure out, could we cluster and identify those? And that's an ongoing collaboration that I'm hoping I can talk more about at some point in the future. But I just want to highlight this method. I think it's really interesting, and it's really a way to sort of go away from sequence-based clustering to, what is the actual 3D surface of the protein. So lastly, I'll talk about structural modifications. This is going to be really quick and simple. It's something we're still building out, but of course, we have both in workflows, but also in our 3D UI, the ability to do single or multi-point mutations. So I can take a surface like this and say, oh, I really think this is, I can see a very big blue patch, and that could be a potential problem that I want to try to mitigate. And so I can zoom in and I can see underneath here is a lysine at the center of that very blue patch. It's hard to see in this image potentially. But so I can take that lysine, I can make it a leucine, and now you can see that when I make that mutation, that very large blue patch gets broken up, and that's maybe desired or not. This is a very sort of abstract example, but something I wanted to show. So you can do that in 3D, you can do it in workflows. You can even in workflows do it programmatically if you just want to explore different mutations of different areas and regions. So part of this is really our modification tools. In addition to this, of course, once we've done these types of changes, we have structure optimizers. You can run MD again if you want to, but you can also actually go in and replace entire loops. So instead of just doing a single point mutation, you can take a CDR and change it completely into a different sequence. It can even be shorter than it was originally. That's the main crux of what I wanted to present today. I'll just acknowledge OpenEye leadership, Anthony Nichols, Jeff Skillman, and Bob Tolbert. I want to highlight our collaboration with Frank Erasmus and Andrew Bradbury at Specifica. And then I really want to highlight John May and Addison Smith in my group because they really do most of this work. I think I have done a little bit of it, but I wanted to highlight them because they really have been pushing hard to get these tools into customers' hands. And then I want to, of course, thank the Orion Cloud team and internal and collaborators as well. So in summary, I'll just say, with this package, and some customers have already gotten access to it, you can now analyze sequences and select diverse hits with high affinity. That's with AbXtract. You can now generate structures of these sequences, prepare them, annotate them, generate structural ensembles, calculate properties and surface properties on those structures, modify those structures, and also simulate the structures and assess their energy landscape and stability. And there really is a lot more coming, and this is hopefully the first talk in another I'll give later this year. So I'll thank everybody for their attention, and I think we'll maybe move on to questions, Janice.
Yes, we are ready for questions. Thank you very much. First question, how does the accuracy and reliability for brand new antibodies, so these with no known 3D structure, what is the validation and testing for such a case?
I mean, I think I'll defer a little bit to some of the validation work that's been done by Charlotte Dean's lab when they built ImmuneBuilder 2 or ImmuneBuilder or Antibody Builder 2, because it is fair to say that we don't really know because there is not a lot of structure and there is an enormous amount of sequence space. At the same time, the validation I think looks pretty good. I think it's HCDR3 backbone is around 2 angstrom RMS to known crystal structures from the moving from training to test and validation. And in some ways, I think that's pretty good. I don't know if moving to 1.5 RMS is really much better. In the same way that I think, unless we really take into account whether these have packing residues and all these other things, I don't think we know. I'll say I don't think we if you have a brand new sequence, I don't know what the confidence level is in that structure. But at the same time, it's a really limited structure space. So it's not like something completely unexpected is going to happen, I think. I mean, of course, there are very long loops. They'll be very flexible. But at the same time, it's not an unending sampling space, right? There are limitations in the 3D structure that dictate sort of the phase space that you can sample. So while I don't know if it's 2 angstroms, 1.5, or 3 angstroms, I also don't know that it matters if you're going to do simulation anyway. And if you don't know what the structure really is in the presence of the antigen, which is probably some sort of induced fit binding. I'm not trying to not acknowledge that I think there is a gap here, but I also don't know that we should be too concerned with the methods that we have today.
Thanks very much for that response. Next question relates to a comment you made about therapeutic antibodies tending to have shorter CDRH3s relative to standard human antibodies produced in a person's body. And the suggestion is that maybe because a lot of the therapeutic antibodies are derived from mice, and mice have much shorter CDRH3s than humans or many other species. Is there evidence that you know of that long CDRH3s, antibodies that have those, have poorer developability?
I don't think I have evidence. It's something I'm certainly interested in. I mean, you might be right that that's why we have shorter, H3 CDRs. I don't know if I think there could be other reasons for that. I don't know that we know that there's evidence for this. At the same time, because what we're really focusing on is what does approved therapeutic antibodies look like? And so what we're just saying is the things that have been approved today, this is what they look like, and they tend to be on the shorter side. That doesn't mean, and I think this was the same with Lipinski's rule of 5, it was a guide. It wasn't meant to be like you have to look like this to be approved. And then some companies took Lipinski's rules too literally, and they stopped developing, molecules that violated 500 molecular weight and 5 hydrogen bond donors or something along those lines, and it pretty much boxed themselves in for years because they thought this is where we need to be. When in reality, we've seen since that there are great small molecule therapeutics outside Lipinski's rule of 5 that have come on later on because you can mitigate some of the issues that come from larger molecular weight or different properties. So I don't necessarily think that you can't develop an antibody that has a long HCDR3, but we just don't know at this point. I think is the real point here.
Next question: could you also compare to hydrogen deuterium exchange experimental data?
Oh, I might meet the context of that question, but I mean, using HDX data, I think is relevant. I know it's for a couple different properties that we use, HDX becomes important. You could of course use that to investigate the antibody antigen interaction if you have if you're trying to mitigate or understand the complex formation of either what is the epitope and the paratope. I know for enhanced sampling simulations, instead of going for normal modes, you can actually give it HDX data and have it drive. So if you had your antibody and your antigen, you can use HDX to drive the simulation to form sort of the initial encounter complex and then the relaxed protein interaction complex. So I don't know when the question was asked, so I don't know the exact context of what it was asked in, but there are different ways we could use HDX, and it is an interesting experimental technique for sure.
Thanks very much. Next question certainly relates to something you were discussing during the presentation, and that is specifically, how do you take care of the dynamic nature of the pocket during pharmacophore calculation? So I should, they're not pharmacophores, and I should clarify that. They're really just colors based on,
negative charge, positive charge, hydrophobic. They do mimic pharmacophore. So I don't know. Maybe it's a semantic difference. But one of the ways we will probably do that is just to take snapshots of different from the simulation, either of cluster centroids or regular snapshots, and just screen these across the database when we cluster. Because I think there's no real way to aggregate that type of structural data into a single query. We could try, and we may end up doing a decent job, but at the same time, this technology is actually incredibly fast. It runs on GPUs, and it's something we can do in a short amount of time. It actually runs CPU and GPU depending on your use case. What we use it for protein ligand small molecule pockets, we can screen 200,000-300,000 patch databases in minutes on a GPU. It takes longer on a CPU, of course. But even here, with these sort of limited clustering type analyses, these are not long calculations, and so we could just incorporate multiple snapshots, both in the database, but also if you think of it that way. But we could just incorporate multiple snapshots, and I think that's probably the way we would do it.
Next question: can Orion software models simulate induced fit antibody binding? That is to say, in parentheses here, changed configuration when it binds to an antigen, for example, in some agnostic antibodies?
So probably, the question really comes down to what do you want to simulate, right? Because I think if we simulate the antibody by itself enough, we could probably get to the configuration somehow of the bound complex. At the same time, it's very much probably an induced fit interaction. So while we could drive a simulation to some, if we just simulate enough, the presence of the antigen really does matter. And we can do antibody antigen simulations. And like I said, with weighted ensemble simulations, there is a way, and I don't think it's something we have ready to go right now, at least not for this context, but you could have two protein structures in your solution, in your system, and you can drive them together. It's really about how much time do you really want to spend, right? So I think Lillian Chong's lab at University of Pittsburgh has done some amazing work on Barnase Barstar, showing that you can drive these two together and get the actual configuration you want. But it's really a matter of how much time do you want to spend simulating getting there. And I think there are ways we can be clever about reaction coordinates and other things using weighted ensemble simulations to get to that point. It's not something we have like a point and click solution ready to go today, but it's something we have been running internally for different interesting properties.
Next question: do you also have plans to extend the technology from RMSD based to energy based?
I don't think we have anything that's actually RMSD based. It's a metric we of course use to estimate structural diversity, but we don't necessarily use it as it's not a driving force by any means. So it's really just a general analysis tool we stick in, and everything we do in this area is really energy based at the core of it.
A couple of things on expansions. And do you have plans to expand formulation suite to biologics?
Not in the next 3 months. But of course, this is an important avenue. And I think some of the tools, I think compared to small molecule drug discovery, where it really is two different teams handling sort of hit finding, lead optimization, and then you hand it off to the preclinical and formulation manufacturing teams. Those teams are very distinct. There may be a couple of people who sort of overlap. I think for antibodies, and someone will have to just correct me if I'm wrong, but my impression from talking to customers and people who do this for a living is it is more of a fluid transition. You actually start thinking about formulation properties much earlier in your development pipeline. That's why developability is a real issue. And you do that, you solve a lot of things in optimization to not have to solve them in manufacturing. That doesn't mean there isn't a manufacturing component. So I'll say, yes, we're absolutely thinking about developability type scenarios, and hopefully some of those will lead into a formulation to help solve some problems in formulation. Whether that'll be in its own suite or that's a good question.
In terms of the molecules, are you planning to support other antibody therapeutic formats such as scFv or VHH?
Yeah, I think so. I think OpenEye has historically not been working on antibodies very much, or protein structure in general. So this is sort of our initial offering to have some things for the customers who have been asking to say, hey, we really love AbXtract, but we really need structure to come next. And so this is our sort of initial offerings to get some tools into our customers' hands, and hopefully for the broader community to see some interesting things here as well. We have a lot of good ideas. How they get prioritized, I don't know yet. A lot of it will come down to, helping customers solve their problems. But VHH, of course, is interesting and a hot topic right now. Single chain FV is as well. So yes, I imagine those will come as well in short order.
Very, very practical question next. Is there a firewall to ensure potential therapeutic candidates are not released into the public domain?
I won't go into the security of Orion. There are white papers, and I'm happy to provide that. We segregate customers very distinctly from each other. They can't see each other's data. They can't access each other's data. Some customers run on their complete own stack. So in terms of network, they're completely segregated from everybody else as well. So we have a broad offering in terms of security, but in all instances, there is no sharing of customer data. So, there is no, I'm sure we get penetration tested and other things like that, like every other company out there. If someone's interested, email us. We're happy to provide white papers on Orion security.
Next question, more technically based. Is there an Orion tool that would allow one to dock the predicted antibody structures to their antigens?
There isn't today. We've talked through a couple different scenarios. I think protein docking is an incredibly hard problem. And I'll just say I don't think there are great solutions out there today. Because while we are developing tools internally, we also, of course, look to the broader community of, where are we at? And it's not that we haven't tried really hard to do protein docking as a field, but I don't think we're nailing it just yet. And so we don't want to bring a tool, like we are developing some tools internally, but we don't want to bring a third-party tool in that isn't very good. And I'm not trying to be rude or anything to the community at large, but it is an incredibly hard problem. And I think historically, if you look at the data, it is just not a solved problem. So could we go into something like AlphaFold multimer? Maybe. But it's an interesting balance, right? Because I want to give customers tools, but at the same time, we do like to do some validation internally of like how good are these tools really. So I think before we do something like AlphaFold multimer, we would probably try to do some internal validation before we do that to see. Because when I talk to customers, some of them say, oh yeah, this is really good. And others are like, well, it really doesn't work for antibodies. And so I need to understand myself, and my team needs to understand, not that anybody's necessarily right or wrong, but on what's the spectrum where this works and what's the spectrum where it doesn't. So we are planning some things, but I think we're still doing some internal validation before we are willing to put this in customers' hands. But that said, we could put AlphaFold 2 multimer, ESM multimer in pretty short order if someone was like, hey, we really need this and we need it fast. And then it's up to them to do the scientific validation. But generally, before we sort of endorse something and put it in, we really do like to do some internal validation first.
Next question is very Very specific. Why was a 10 nanosecond simulation run in MD simulation, while in enhanced sampling, it was extended to 3.5 microseconds?
That's a great question. I think maybe I didn't sort of couch this properly. So when we do 10 nanoseconds, we run a single simulation out for 10 nanoseconds. When we do weighted ensemble simulations, we define a reaction coordinate, and these are the normal modes. And the way enhanced sampling works, without getting into the technical weeds of it, is as you move along that reaction coordinate, you sort of split your simulation to say, hey, this one made progress. We should make more of these to see if it'll make further progress. And so you're sort of propagating along these normal mode axes. And so you're simulating at each bin along that axis. And you're doing multiple simulations in each bin potentially. And you're accounting for the probabilities as it's moving back and forth across these bins. That's how we sort of adjust the kinetics and can assess the rates and other things and the energy landscape. So at the end of the day, we do very short simulations in each of these bins, and we keep doing them for an extended period of time. So really what we're doing here is more exploring the phase space versus relaxing the structure. And 3.5 microseconds is just what it comes out to when you do 50 iterations of WESPA on Orion with the chosen reaction coordinates. It's not a hard number. It's not like we chose to stop at 3.5. It's just after, I think, 25 or 50 iterations using WESPA as a technology along these specific reaction coordinates, that's what it came out to. And so it's a little bit of a technical question. I'm happy to take that offline as well. I think David Lippard has done some really nice presentations. I know there will be a webinar from him if there isn't already at Eyes Open or at OpenEye on our website, talking about protein sampling, which I think we'll sort of discuss this in much more detail.
Okay, I think we have time for another question, maybe two. The question is, how do the antibody builder structures compare to AlphaFold?
I don't think I know the answer to that question directly. I know when ABodyBuilder 2 was published, they sort of looked at the state of the art to say, you know, where is the state of the art and are we comparable? And I think they were. I forget exactly what they compared against, but AlphaFold 2 was one of them. I actually think maybe ImmuneBuilder or ABodyBuilder 2 is slightly better than AlphaFold, at least in the version they were testing against. And ImmuneBuilder, to note, you know, was sort of built on the AlphaFold technology, but instead of training on the entire protein space, the known protein space, they focused the training really around what does antibodies look like. And so in some ways, it's a fine-tuned version of AlphaFold for antibodies.
Okay, if you can answer this last one in 30 seconds.
That's fine.
Well, go ahead and ask it. Can you expand where we can find algorithms used for developing software?
For Orion, we have public documentation about how to put things into Orion. And in general, we look for things, you know, in GitHub, we look at the license terms and other things like that. And I think that's the short answer there. How do we use this software? You would contact our sales and support team, and they try to get you onto Orion.
Perfect. Excellent. Thank you so much.
All right, thank you.
A wonderful presentation, but also for giving all of these insightful answers. So I do here definitely want to thank Dr. Sorensen for providing these insights into 3D antibody modeling in Orion. And I thank all of our attendees for coming today and joining in the webinar. An on-demand version will be available very soon, and I'll send a link to it via email to everyone who registers, so you will get that. Please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.
Thanks, everyone.